This is the second video of the Mouse Genetic Approaches for Neuroscience lecture. In this video, we will talk about genetically modified animal models with focus in knockout and knock-in animals and how they're different from transgenic animals previously described in video number 1. Knockout animals are animals whose DNA has been genetically engineered so that it does not express a particular protein. The main difference between knockout animals and transgenic animals is that in the case of knockout mice, it's required targeted mutagenesis. So we specifically target the transgene to a particular place in the genome. And targeted mutagenesis is achieved by homologous recombination, a concept that we will explain in a minute. Knockout animals are commonly generated using DNA transfer and embryonic stem cells implantation, as we described in video 1 for the generation of transgenic mice. The technique is the same. The main difference is that in the previous case in transgenic mice, the insertion of the cassette is random in the genome. In this case, we used homologous recombination to target the transgene to a specific place in the genome. For this, we choose a targeted site within the gene. In this case, will be in the middle of this gene X, which is the target gene. What we do is we generate a synthetic cassette containing an antibiotic-resistant gene that is flanked by a 5' and a 3' region that is homologous exactly to the place in which we want to insert this entire cassette. Then when we transfect the embryonic stem cells with this transgene, occurs a homologous recombination, and then this neo-cassette gets inserted in the targeted position in the transgene X, and now the gene is interrupted. So this causes an interruption of the gene, and the target gene does not get expressed anymore. So this method can be used, for instance, for gene therapy to correct the pathogenic mutation by restoring the normal DNA sequence. It can be, so if there are specific mutations in a particular gene, like for example in this position, that could be removed by this technology. In this case, this is a more advanced cassette to help you select for those ES cells that have been effectively transfected. In this case, what we do is we use a 5' region that we want to use for homologous recombination, and we use a 3' region for homologous recombination. So anything that is between the 3' and the 5' region will be removed and replaced by our construct or transgene. We choose to remove the exon 2, that's what the E stands for. We choose to remove exon 2 from the target gene. And we generate a construct in which there is this 5' homologous recombination region followed by a neo-cassette, which is an antibiotic resistance cassette that allows selection for cells that incorporate the target construct. And whenever the cells are transfected with this cassette, this confers resistance to neomycin analogs. And cells with neo will die, cells without neo will die in the presence of neomycin. Then the construct will be followed by a 3' region that is after the exon 2. And then we will also use a TK cassette that stands for thymidine kinase. And this cassette allows for negative selection for cells that have not undergone homologous recombination. Because when there is a homologous recombination, the TK cassette will be removed from the transgene, and only this product below is what gets inserted into the genome. But if the transgene gets inserted somewhere else by accident, not using this 3' end, then the TK cassette will still be present. And the presence of the TK cassette confers sensitivity to ganciclovir. So when cells are exposed to ganciclovir, if they still keep the TK cassette, they will die. So then we use a positive and a negative selection to ensure that the cassette is being inserted in the right place. So a very famous technique that has appeared in the last years to generate knockout animals is the CRISPR-Cas9 technology. And CRISPR stands for Cluster Regularly Interspaced Short Palindromic Repeats. And this system, it uses the bacterial immune system that has been modified for genome engineering. So largely due to its simplicity and adaptability, CRISPR has rapidly become one of the most popular approaches for genome engineering. And the CRISPR-Cas9 technology uses two components. One is a guide RNA that guides the Cas9 to a specific target and position in the genome. And then we use a non-specific CRISPR-associated endonuclease, Cas9. So the way this works is we will design a specific guide RNA for a position that we want our Cas9 to be directed into the genome and cut the DNA. So this technology is used for generation of knockouts, also for gene repression or even gene activation. And the Cas9 that is most commonly used is derived from an organism called asparagines that recognizes a PAM sequence that is specific with this nucleotide composition. And there are different endonucleases or Cas nucleases now available for generation of knockouts using CRISPR. So this is a detailed mechanism of how CRISPR-Cas9 works to cause genome editing. And in this case, as I mentioned earlier, we will use a guide RNA which has a scaffold and defined sequence, and then we add the spacer or target sequence that we want to direct our guide RNA to. Then we will mix that with our Cas9 enzyme and the complex forms. So there is a guide RNA Cas9 complex that forms, and then there is a target binding. So the Cas9 is directed by the guide RNA to the target site in the genome. And then there is a recombination binding between the spacer and one of the strands of the gene, and that causes a cleaved double strand binding, double strand DNA. Then this cleaved gets repaired, and this can be repaired by different mechanisms. If the cleaved gets repaired by non-homologous end joining recombination or NHEJ, and then there are a series of different modifications that can happen in the genome, and this is completely random. There could be an insertion, there could be a deletion, or there could be also a frame shift. In some cases, it could be perfectly repaired, so no modifications have been added into the genome. In this slide that I'm not going to read, but you guys can take the time to read it and see all the details about CRISPR-Cas9. Not only this slide provides details about the recognition sequence of the guide RNAs and the Cas9, but also the different DNA repair mechanisms that the Cas9 uses for genome editing. We just described the non-homology and joining repair pathway, and there's also details for an additional DNA repair, which is the homology DNA repair mechanism that will be used in the next slide to introduce gene mutations. You can also find in Canvas an optional video that very well describe the CRISPR-Cas9 genome editing. CRISPR-Cas9 technology can be also used to generate point mutations. In this case, the DNA repair mechanism that Cas9 system uses to introduce point mutations is called the homology directed repair, or HDR. HDR can generate specific nucleotide changes, ranging from a single nucleotide change to a large insertion, like the addition of a fluorophore or tag. For this technology, we will need three elements. We will need the Cas9, we will need the guide RNA fused to the spacer, which contains the target sequence where we want to make a point mutation or introduce any additional element. And then the third element will be a template repair. This template repair must contain a desired mutation or a point mutation, and it has to contain also additional homologous sequences that immediately upstream and downstream of the target. The length of each homology arm is dependent on the size of the change being introduced, so with larger insertions requiring longer homology arms. The system and the process is similar to the one that we just described in the previous slides, but in this case, once the Cas9 identifies the target sequence and generates a double-strand break in the DNA, then the HDR system uses the template repair or the repair template to fix this double strand break and uses this to introduce this new repair template in the cut position. And now we've just generated a precise edition. So now we will talk about knocking mice. And basically, the generation of knocking animals is just a variant of the knockout procedure. And here what you can see is the same construct that we used to generate knockout animals, but in this case the target endogenous gene is itself designed to be expressed under the control of the promoter elements of the knocked out gene. So basically, we will be using the same construct as we described earlier, in which the homologous region recombination 5' will contain the promoter of the gene A, which is the gene target that we want to remove. And then we will use as a region 3' the exon 2 of the gene A. And anything that is contained between the 5' and the 3' region will be removed. Then what we will insert in the middle is an additional gene. Now we call it gene B, and also a neomycin cassette for positive selection. We can also use the TK gene within our cassette to ensure a negative selection. And then the result of this recombination is that we will keep the promoter of the gene A, and that will be followed by gene B that now will be expressed under the control of the promoter A, and will be followed by a stop codon. Then that will continue with a neomycin cassette that will confer resistance. And then the chimera exon 2 from the gene A that will not be expressed due to the presence of that stop codon. So by doing this, we just generated a knocking mice that is expressing a gene B behind the promoter of gene A, and then gene A is knocked out. Here there is a short quiz that you can do at home after watching this video. And if you have any questions about any of these, we will also see these questions in class.